Good morning, everyone. As you can see, the title of the talk is different from what announced because I'm a replacement speaker for my CEO, Dr. Ram Prasad. He could not make it today to come here. So I'm Associate Director of Genomic Medicine and Personal Genomics at Medgenome. So I'll be talking about more on the genetic study that we did that Dr. Shweta was mentioning. On the PD genetics, I think it's one of the. It's not only one study, so it has been three studies we did in collaboration with PRI. So I'm not talking more on the PD thing, but what I understand, it's a very complex disease overall in whichever aspect you look at it. For diagnosis, even the molecular diagnosis, it's a very complex disease. And this is the global burden. You can see, like, so this is a 2016 data, but we don't know. So this is the estimated number. So as the previous speaker was telling, it's not very highly prevalent in India, but still it could be. I don't know, I'm just guessing because of the diagnosis, and you showed like more than 100% increase is coming up. So it could be because of the undiagnosed thing in the rural spaces. Ah.
Um, Having said that, the prevalence of Parkinson's disease is low in India compared to the Caucasian countries. It's because of the total population size, we have much higher number of patients.
Okay, as
and
a
we
proportion.
really need to focus on Parkinson's
I
disease.
understand. Okay, thank you. Now this is the disability adjusted life years. So you can see the South Asians are placed here, and you can see the life years are more in the high income countries compared to the low income. So the lower in the blue part, you can see like the African sub regions of the Africa. So we stand somewhere here. So coming to the genetics part of it, genetics can explain as per our present standing. It's up to 25% of the disease can only be explained by genetics from the discovery so far, from the 90 risk loci and 10 to 15 causal genes, and the unknown reasons are almost 75%. So coming to the genetic part, there are two variations of it. So like one is like the monogenic or the causative thing, which is rare mutation, the variations, genetic variations that are found rare in the population, and the second is the polygenic variation. So small effects from hundreds or thousands of genetic markers. So this is the evolution of the genetics part. It aligns with what Dr. Shweta was mentioning. So far 90 risk loci were discovered. In the pre-GWAS era, so isolated genetic studies were there, not like a genome-wide association study, but analyzing individual genes. So from 2005 onwards, the discovery has been increased, but compared to other diseases, I would say it's very limited, like 90 risk loci. And what are the applications? So one is like the discovery, the number of markers have been increasing with the time. The genetic risk loci discovery has been increasing. Now it's 90, and we have few more now. So and there have been large consortia and efforts and all are there. So more discoveries will happen. And this can be brought to the translational, how it can help a patient. So that's the bottom line. So it can help in target discovery. I'll show you one example that in the recent study that we did. So and the discovery of new markers and all. Now, basically, Medgenome was part of this three study published. One is, the most recent is the YOPD genetic study with almost 700 YOPD samples and 1,300 controls. And a pilot study was conducted 2 years back with 100 patients. And a clinical research paper was explaining the clinical characteristic, the non-genetic part of it, of this, the whole cohort was also published. And the PD clinical study, it's a completely clinical study. I'm not going through to read out the points, but basically the clinical characteristics of the 700 patients were described in that paper. And the pilot genetic study, we had, as I told, 100 whole genomes, where whole genome sequencing were done on 100 patients, and we described the data. So we were seeing, though the sample number is limited, 100 patients were there, so we could see, for example, in the polygenic risk score, we could see a difference between the controls and the YOPD patients, and we could see a trend on the, high polygenic risk score for the, for cases and all those. It was a pilot study paper. So we could also find in that paper the copy number variations in the Parkin gene, so in around 10 samples. And the latest PD study we have, we looked at the, we have, around 700 samples, and we looked at the genetic aspect in multiple ways. One is like looking at the rare variants, that is the causative reasons on the diagnostic point of view. So and the other part is like the common variants. So we applied 3 technologies here, like the whole genome genotyping array. In the whole genome genotyping array, Medgenome came up with a proprietary genotyping platform for South Asian population, so which is underrepresented in the genetic research. So we have a product which is to genotype the the variants that are seen majority in the South Asian population plus the common variants. We use that platform, and we have the whole genome sequencing plus the whole exome sequencing was also done. So this is the kind of a data point of view. So which all type of data. On the top of that, we could add 1,300 controls also added from our Genome Asia cohort. So Medgenome is part of the Genome Asia consortia, where we have more than 6,000 individuals, South Asian genomes. So coming to the sample, PD sample summary. So we had a early onset and YOPD majority. We had few LOPD and JOPD individuals, and majority were sporadic PD, almost 80%. And this is I am not going through the detailed data analysis workflow. So basically, I'm a genetic data scientist. So we had to plan the experiments. As I told, it's a multi-technology, multi-platform data generator. So we had to devise the data analysis. It's like may add complexity if I try to explain. So basically, from the Genomide Association study, our major findings were 2. One is like the SNCA loci, which is well known. And well known, when I say well known, it is shown in the more than 1 million samples, 1 million patients in the IPDGC consortia, European cohort. And they were, and in the European study, as the sample size were large, so the significance value is E minus 100, more than 100. But in our cohort, modest cohort, we were getting Genomide signal above E minus 8, and these are like kind of comparing their p-values, the statistical significance, what we got in the prior cohort and what is there in the IPDGC cohort. So we could replicate many of the markers in that loci, and this shows the correlation basically. And interestingly, we found another loci also crossing this Genomide threshold, CCDC C85A, which is a which is not well known in PD, so which need further experimental validation. So we found this signal with just 675 samples in the Indian cohort. And coming to the the rare variant part, so this is basically the mutations that we found in the genes, in gene names you can find in this panel. So mainly we found like the most monogenic mutations were found in the GBA, and then Parkin, and so on. But if you're coming to the variant classes, you can see like the majority, the highest pathogenic variants were found in the Parkin gene, And then a lot of VUS, you can see the background. So there was a discussion in the VUS side. So this is a diagnostic odyssey. It's complicated. Until and unless we have evidence, we can't relate. We can do a molecular diagnostics, to a likely pathogenic or a pathogenic variant. But one thing was there actually. So in the diagnostic one, we could find a genetic variant that is seen predominantly in the Indian population. So that is that genetic variant, monogenic variant, falls in the GBA gene, and a validation study has been done. So on this particular genetic mutation, so serine to arginine 164. So it was done it was done by our collaborator Denali Therapeutics US. So basically, it's a GCase level. So this mutation is similar to comparatively to the knockout thing. So if someone is having this mutation, or the individuals diagnosed with this mutation is like a GBA knockout. So this was shown in triplicates, and also we measured the GCase activity. The enzyme activity was also measured. So it is, as you can see, the enzyme activity is like 0, like a knockout compared to the wild type. If one has, this was in the mouse model, but if one has, we found the patients with this mutation in the Indian cohort. So this is the first evidence that shows this mutation. Probably this genetic variation will be added in the ClinVar database as an entry of a pathogenic or a likely pathogenic thing. Then I'm coming back to another slide again shown by Dr. Shweta. So basically here what we are observing is, as I told, the diagnosis is a challenge actually. In the genetic diagnosis is a challenge. So this is the polygenic risk score for the control group. You can see the median values here. So here on the extreme right side, these are pathogenic and likely pathogenic, where we have an answer. Okay, we found this variant that's a SNCA or GBA, or that are associated, that are not associated, that are causative for the disease. But if you look at the polygenic risk score of the clearly diagnostic cases, it is comparable with controls. But if you look at the VUS or the null cases, these samples have a median polygenic risk score higher than the likely pathogenic or the pathogenic. What does it mean? So it means not only the classical well-known causative mutations, but also the common polymorphisms that act together to confer the genetic risk for these patients. So which are actually in practice now classified as VUS or the null cases. And in the study, we could also do an analysis called gene burden. How much burden for each gene. We quantified it, and as you can see, this is a QQ plot where we can see like the gene that is coming in top is the bazoon gene, and the thing is, like, we did not have the enough statistical power, 700 samples. To come up with a whole genome significance for this gene, but this need further validations and to have validation. And this is already reported in PSP disease, so bazoon gene, but first time showing as a significant one in the PD. So conclusions and future directions. So basically, we found like the major drivers, genetic drivers, causative drivers are Parkinson, GBA, PINK1, LRK2, which are very well known, and Parkinson, a lot of Parkinson mutations that we are finding in the Indian patients, as I was showing in that copy number variation thing. And SNCA is a well-known loci that we identified also in the Indian population, and along with that a novel association which needs further validation, like that CCDC C85A. And polygenic risk score can be used as a good screening tool. I won't say diagnostic tool, a screening tool. So somebody who has a family history or very early symptoms, or anyone can do a screening test to know the polygenic risk score for PD. And I don't know what are the next directions that one can take, but probably some precautionary measures or some lifestyle interventions can be taken. And the gene burden analysis further needs a validation. So it's bazoon gene came up in the Indian cohort here. And future directions also I would like to add, like coming from genomics, next level can be the proteomics part of it, so which can be which can further validations or further proteomic studies can also be done to in the Indian cohort to know the things. We see many things are overlapping with European, but we also see something, some few things are specific also for Indian population when we come to the PD genetics. Thank you.
Thank you, Ramesh sir. We can have 2 or 3 questions.
So this is regarding the Basun gene. I have a naive question. So when you say gene burden, what are you implicating? Is it overexpression of the protein you're considering, or?
No, actually, so how much are the damaging gene, the damaging genetic variants, so the loss of function variant, the missense variants, all those variants are highly, the number of variants, those variants are present in high percentages in this gene, so compared to the other gene. So this is like for each signal is not coming as significant, but altogether this gene has maximum number of damaging genetic variants.
I understand that now. A follow-up question I had was, when you have these variants, do you also go for protein prediction, protein function prediction in terms of let's say dominant negative or something, because the variants may not be it may not be homozygous in.
Yeah, that's the main thing. So like the GBA gene validation that we did, we also need to do validation for the Basun gene. Only thing that we could not take it up because it did not have the genomate significance level because of the statistical power. We also did some statistical power analysis kind of a thing. We need at least 4,000 Indian patients to reach that level.
Thank you.
So it's actually follow-up of Ravi's question.
Yeah,
so
yeah.
Basun caught my attention also. So it's a huge protein, as far I know.
Okay.
So did you see mutation in one particular single SNP, or are there multiple things?
Across.
Is there any particular regions affected, or?
Exon-wise map I don't have now, but it's across the gene.
It's all in the coding region.
All in the coding region.
Okay.
We select only the coding region one, and also have an impact, functional impact in the annotated databases, and also it should be rare. Rare, like less than 1% seen in the population. So these were the criteria qualifying for that variance in that gene.
And nothing is known about the expression in the patients?
This gene is highly expressed in the brain. We checked in the general, I'm telling. So we did not check in the patient's sample cohort we have. Yeah.
Thank you.
This Basun gene, we looked into patients' clinical aspects of that, because Basun was told to be associated with the PSP cohort. And when we checked into the PD cohort patients of ours, most of these patients have gait and balance issues. So given that even in PSP that it is more of a gait and balance related issues, so Basun should be somewhere related to a gait and balance type of a function. That is an reduction. That's it may not be connected also. That should be other way around, because PSP patients may not respond with the dopaminergic going on. And if you look into Basun gene is not reported in the other international publications where related to Parkinson disease. And many of these, when you have young onset PDs and other things, they have primarily gait issues. And there we have found all those Basun mutations have come as positive or VUS.
So then, just to add to what he was saying, in the Drosophila, what we are studying is a neuromuscular junction.
Okay.
So connections through the synapse to the muscle. In those functions also, if you have problems with Basun in terms of its ability to release help in release the neurotransmitters, we see movement disorder-like symptoms in the larvae or the adult fly.
It's a very interesting point. From I'm just Dr. Prashant is the right person. We did not do further investigation on the molecular side or the proteomic side, but we can check the deep clinical data that we have. So to see whichever patients that we are seeing Basun gene mutations, so we can identify the burden carrying individuals, whether they have this kind of symptoms significantly compared to the rest of the group. This can be just based on the clinical data that we have.
And what we generally do is that if those variants, if they're conserved, let's say in Drosophila or in C. elegans, you can then try to make a knock-in variation of the variant in that organism to see if it phenocopies movement associated or
Okay. Yeah.
tremor associated gates. Those are easy. Good.
Just one question I had, whether this bassoon and the alpha-synuclein, are there any correlation? Do you find them existing together?
We did not check that.
Not checked, okay. Thank you.
Bassoon, we did not proceed further just because if we do further experiments and validations, we won't be able to justify the statistical significance. Rather, we showed the other way, like we showed in one figure, we showed we don't have enough statistical power. So we need 4,000 samples to get there.